Today I'm going to give you the most popular lecture I give. This is the lecture that everybody seems to like. So I hope you like the lecture today. If not, maybe I don't stop giving it. But this is a very popular lecture because it relates protein structure that we've been talking about to physiological function. And I think with this lecture, for the first time, you can start to see why considerations of protein structure are really important as regards your body. Okay? So I start this lecture by talking about, last time I finished, in fact, I changed this slide to correct this slide from yesterday. Oh, wrong one. The slide from yesterday that had the forces. Okay? So that should have said disulfide, and so I've updated that. So that now says disulfide. The difference between a disulfide and a sulfhydryl is a sulfhydryl is an SH, and a disulfide is an SS. So you can see this is an SS bond, two sulfurs joined together. So I wanted to make sure I got that corrected for the thing. All right. Now, today what I want to start talking about, I'm not going to talk about the techniques. You're not going to be responsible for that. There are various ways that we can determine protein structure, but I think it's really beyond what we need to consider in this class for how that happens. It's much more fun and much more interesting to consider how protein structure actually relates to physiological things, as I said. So one of the proteins that's very interesting in this respect, I talked about a little bit yesterday, is myoglobin. And myoglobin, as I mentioned, is a protein. It's related to hemoglobin. And it has the same functional group. That's just simply something that's attached to the protein. It has the same functional group as hemoglobin does, specifically the heme group. And the heme group, I mentioned to you yesterday, was a fairly flat, planar structure. Okay? It's fairly flat. And this is shown right here. This is what that complex, what that flat thing that you saw in the structure yesterday. And if you're wondering what that structure was, I'll show you briefly. That structure was right here. Okay? So right there, this sort of ring structure here is the heme. Below it, I said, was the oxygen. And if we consider that heme structure, this is what it looks like. And we can see that it's a big old ring structure that's there. And in the very middle of it, we see an atom of iron. Okay? This structure turns out to be a pretty interesting structure. Protoporphyrin is the thing without the iron atom in it. If I put a magnesium in here, I end up with chlorophyll. Okay? So chlorophyll and hemoglobin have almost identical structures. The difference being that chlorophyll has a magnesium in that place instead of iron. Okay. Well, plants don't use chlorophyll for carrying oxygen. Plants use chlorophyll for capturing the energy of the sun. We use the heme group along with the iron to carry oxygen. That turns out to be very important. The myoglobin that does that is a protein, and it's not a protein that's really good for carrying oxygen. Myoglobin is very good for storing oxygen, but it's not so good for carrying it. We'll see why that's the case in just a little bit. Okay? It's very good for storing it. Okay? So I like to think of myoglobin as something like a battery. All right? So if you have a laptop and you're doing some very critical work, okay, and the power goes off, the laptop continues to function, and the reason it continues to function is because it's got a battery that steps in immediately and replaces what was coming out of the electrical circuit in the wall. Okay? That battery fills in the void. Well, myoglobin fills in the void too. Myoglobin is a protein that's found abundantly in our muscles, and it stores oxygen so that when oxygen gets very low, it gives up oxygen so the muscles can continue to produce energy at a very efficient level. Without oxygen, muscles don't work very efficiently. Okay? So having this sort of oxygen backup in the form of myoglobin is really helpful for muscle cells. Okay? Well, this shows the ring of the heme, and there's the iron atom that's there. And what you see here on the right is what happens when myoglobin or hemoglobin, what you see on the screen happens for both myoglobin and for hemoglobin. Okay? What you see is what happens when that oxygen binds. And we saw that oxygen binding in that figure I showed earlier. with the two red spheres that were there. And that's the two oxygen atoms of a molecule of oxygen, as you can see right here. It's being held in place by the iron atom. Now, if we look over here, we see something that's not oxygen. In fact, what we see over here is carbon monoxide. And it turns out that carbon monoxide has almost exactly the same shape as molecular oxygen. So carbon monoxide can bind right there and replace molecular oxygen. Okay? One of the reasons carbon monoxide is poisonous to you is because it can replace oxygen in hemoglobin, and your body doesn't get enough oxygen. You can actually suffocate like that. Okay? There's other things that kill you with respect to carbon monoxide, and we'll talk about those later also. Okay. Let's see, where was I? Right here. All right. Oh, let me come back to that. I'm going to come back to that and talk about that in a bit. Now I want to turn our attention to hemoglobin. So I've started talking about myoglobin. Let's talk about hemoglobin. Hemoglobin is related to myoglobin, but there's a very big difference. Okay? Now, you can't really tell just by looking at this because it doesn't tell you, but the fact that there's two different sets of colors on here should tell you something. It tells us that there are multiple subunits here, multiple proteins being held together. It turns out there are 4 of them. Myoglobin only had 1. The 4 subunits that are found in hemoglobin are all very closely related to myoglobin, but with some important differences. Okay? Each of the subunits, 2 red and 2 blue, each of the subunits has a heme group. You can see 1 heme group back here. You can see 1 heme group here, 1 down here, and 1 over here. Okay? So there are 4 heme groups that are present, 1 for each of the subunits present in this hemoglobin. The 2 blue subunits are identical. They're known as alpha subunits. Okay? And the 2 red subunits are also identical. They're known as beta subunits. So we refer to hemoglobin as something that has a structure of alpha 2, beta 2. Okay? Alpha 2, beta 2. Now, because hemoglobin has a multiple subunit structure, there's some very interesting properties that the molecule, or that this protein actually has. All right? So let's start with oxygen binding. Okay? Now, I'm showing you a plot of percent saturation versus oxygen pressure. By the way, if I ask you to draw a graph on the exam, okay, your exam will automatically be counted wrong if you don't label your axes properly. Because without proper labeling of axes, a graph has no meaning. Okay? So it's important that you label axes properly when you're drawing a graph. Otherwise, the graph is just a squiggle, and squiggles don't tell us anything. Okay. Now, I say that because students tend to remember squiggles much better than they remember labels on graphs. Squiggles don't mean anything unless the graph is labeled. Okay. Now, so the y-axis here, we're plotting percent saturation. That is, what percentage of the molecules that we have in there are loaded with oxygen. On the x-axis, we're plotting the pressure of oxygen. You can think about it as the concentration of oxygen if you'd like to. Okay? Well, what do we see? We see one plot for myoglobin, and we see a very different kind of a plot for hemoglobin. Okay? Now, what I've told you so far, we can actually explain with this graph. What I've told you so far is that myoglobin is not very good at delivering oxygen. And that's shown in this graph right here. What does this graph really mean? Okay? Well, let's look at it. We see that up here at reasonably high concentrations of oxygen, myoglobin has about 100% of its subunits holding onto an oxygen. Well, that's good. You want that, right? If we start decreasing the concentration of oxygen, we see that myoglobin holds on to oxygen very, very well, getting way, way down here. Okay? So we're down here below 10. We were up here at 50, and we still have probably 80 to 90% of the oxygen. Well, that's not really good, because the concentration of oxygen that's in our lungs is about out here somewhere, where these two lines actually will come together. Okay? It's about out here. All right? We want myoglobin to be able to hold on to oxygen, but if we were using myoglobin to deliver oxygen to our cells, it wouldn't let go of oxygen unless the cells were really starving for oxygen, which is way down over here. Okay? Now, myoglobin does give up oxygen to muscle cells, but it only does it when the oxygen concentration gets really low, way over here. When the oxygen concentration is very low, then myoglobin starts dumping its oxygen, and that's to keep the muscles working efficiently, because they work much more efficiently when they've got oxygen. Okay? Well, let's look at what hemoglobin does by contrast. By the way, this graph is called a hyperbolic graph. That's the shape of the curve. It's called a hyperbolic graph. Okay? Hemoglobin has a curve that we call a sigmoidal graph. And sigmoidal is like an S. There's the lower part of the S, upper part, and then it curves over like this. Okay? If I were to have a little bit higher concentration of oxygen, what I would see is that these two lines would essentially meet up here somewhere. And the concentration of oxygen in the lungs is about up here somewhere. What this tells us is something kind of cool. It tells us that hemoglobin, at very high concentrations of oxygen, such as we have in our lungs, can get completely saturated, that is, loaded with oxygen. And as it leaves the lungs, where the oxygen concentration starts falling, so too does the amount of oxygen that hemoglobin holds. It starts falling as well. So it doesn't have to get all the way to the place where the cells are starving, which is over here for oxygen. It can actually start giving up oxygen when cells are really starting to need it. Oxygen's very good in this flexible way in which it delivers oxygen. Okay? Now, I'm going to explain to you at a protein level now what actually happens with this different affinity that hemoglobin has compared to myoglobin. Okay? So let's think about that. Oh, sorry, wrong one. Okay. So let's go back to the structure here. Okay? Now, I said this was true for myoglobin. It's also true for hemoglobin. Okay? The structure, and actually let me go back even better. Let's go back to the actual myoglobin structure here. So if we go here, okay, this is a subunit of myoglobin. We can think about it just like it's one of the 4 subunits of hemoglobin. Okay? It's going to behave very similarly. All right? Now, we've got this heme group right here. For the moment, we're going to imagine there's no oxygen in there. Okay? The heme group, as I said, is pretty close to being flat, but it turns out it's not exactly flat. Okay? Now, if I had my 3D classroom with the goggles on, I could actually show you this, which I don't have at the moment. But as I said, sometime I'll show you guys that. With the 3D goggles on, you can actually see the shape of the ring and all this sort of stuff in 3 dimensions. So you actually can not try to imagine it, which is what I'm going to tell you right now. Let's imagine my hand is that plane of heme. And in the middle of my hand is where that iron atom is. Okay? Now, you have to look at this very carefully. I'm going to exaggerate it with my hand so you get a better idea about what's going on. All right. What we have is my hand, and the way that it's drawn is exactly where my hand is. The oxygen will come up under here. My hand is here. The iron is attached to an amino acid called histidine. It's actually linked to an amino acid, and that's what you see part of right there. The amino acid is linked to the entire rest of the protein. All right? All right. Here's my hand. Here's the heme. Here's the iron. Here's the histidine that's up here that's holding on to it. Everybody got that in vision? Before the oxygen gets there, this flat ring is actually a little bit concave. It's a little bit like this. I'm exaggerating it, but that's about what it looks like. It's concave. The iron is up at the very top, the highest point, and this thing looks kind of like this. When oxygen comes in, so when the hemoglobin enters the lungs, oxygen comes in and it goes bloop right up here. And when it goes bloop, what happens is the iron atom moves slightly down. Everybody see what happened? Oxygen comes in, the iron moves slightly down to get closer to the oxygen. It's a very, very tiny movement. It's a fraction of an angstrom. Okay? That very tiny movement is responsible for you being able to be alive as an animal. I'm going to explain why in a minute. But that little tiny movement is why you are able to exist as an animal. All right. I'll tell you why that's the case. Well, it takes me a minute. Okay. We've just seen a slight movement. We've seen that the iron atom has moved a fraction of an angstrom down. The iron atom was attached to a histidine, and the histidine is attached to another amino acid, which is attached to another amino acid, which is attached to another amino acid, which is attached to the entire rest of the protein. The toe bone is connected to the ankle bone, the foot bone is connected to the ankle bone is connected to the knee bone. If I pull on the toe bone, what's going to happen to the leg? It's going to move. Right? I'm going to see it move when I pull on the toe. I've just pulled on the toe, this toe being an atom of iron. Oxygen has pulled it down, and as a consequence, the shape of this entire subunit has changed very, very slightly. Okay? You would have a hard time looking at it and see the slight change that it existed. Even at this magnification, you would have a hard time seeing the difference. But nonetheless, it changed shape. Now, in myoglobin, this has no effect whatsoever. No effect. It doesn't change anything about myoglobin except for that very slight shape change. Hemoglobin is different. Hemoglobin has 4 subunits. When I change the shape of one of the subunits, it's interacting with 3 other subunits. That changes the nature of the interaction of that first one with the other 3. Okay? That little change causes those other 3 to also change. Now we're starting to see some little very tiny thing that's essentially being magnified because we're starting to see changes across all 4 subunits that's happening as a result of the change that happened here. Those other 3 subunits now change from being in a state where they don't like binding oxygen to being in a state where they do like binding oxygen. All 4 start out, they don't want to bind oxygen. It takes one to gobble up an oxygen, and then magic happens. The magic being that all 3 other ones change because of the thing I just described to you here. Well, this turns out to be really important because hemoglobin is traveling through our lungs pretty quickly. We want it to get as much oxygen as it can as it's traveling through there, so we change its binding affinity, that is, the desire to bind to oxygen, and all of a sudden, the other 3 subunits go gloop, 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 gloop. That was 4 gloops, but 3 gloops, okay? They actually bind that oxygen, and they leave the lungs loaded with oxygen. The binding of the first oxygen increased the affinity of hemoglobin for the other 3 oxygens. The binding of the first oxygen by the first subunit of hemoglobin, it doesn't matter if it's alpha or beta, this happens for both of them, it doesn't matter. The first one that binds an oxygen changes the other 3 and increases their affinity. This phenomenon I've just described to you is called cooperativity. Cooperativity. Cooperativity occurs when the binding of one molecule to a protein increases the binding of others of the same protein. Okay? That's cooperativity. Now, I said that was essential for animals. Why was that essential for animals? Okay? Well, animals need a lot of oxygen, and they need to be able to get it very quickly. Animals use oxygen in their muscles to make energy. Plants don't have muscles. Because plants don't have muscles, they can't run away. If something is chasing them, or if they are chasing something, animals have very drastically differing oxygen needs. When you're being chased by something, you want to get as much oxygen in your system as you can. You start breathing heavily. Okay? To deliver that, your body starts using oxygen. You want to get it working very quickly, very efficiently. Cooperativity makes it possible for you to do that. If you didn't have cooperativity, you couldn't have muscles. Muscles are necessary for animals. Okay, so that little thing you saw right there, that little tiny change that you saw, was necessary for an animal to exist. Okay? Structure really is important. Structure really is important. Well, when I talk about structure, I neglected to mention this structure right here, quaternary structure. That's the last level of protein structure. And it arises when we have multiple subunits that interact. That's what hemoglobin is. Hemoglobin has quaternary structure because it has multiple subunits that interact. Myoglobin doesn't have quaternary structure. It's only got one subunit. Okay? Many proteins have multiple subunits. Many proteins, therefore, have quaternary structure. All right. Well, this is pretty cool. So now we start to see how oxygen comes in and binds to hemoglobin. We see how cooperativity allows us to load up the hemoglobin that we need for our muscles, especially as we're trying to get it loaded up as quickly as possible. All right? Now, this graph shows us that as the oxygen concentration starts falling, hemoglobin starts releasing oxygen. That in itself is pretty cool. But that's not the whole story about how hemoglobin works. Okay? That's not the whole story about how hemoglobin works. Keep in mind that we have different tissues in our body that have different needs. Very deep in our muscles might be very different than the needs of, for example, our liver, or the needs of our skin, or the needs of other types of muscles. Okay? So we have intestinal muscles that are different than muscles that are used for running and jumping and so forth. Okay? What we really want is to be able to deliver oxygen as quickly as we can to the tissues that need it the most. Well, this works nicely on an average. I'm sitting around watching the tube, and my oxygen needs are sort of constant through my body and so forth, right? But if the house catches on fire and I've got to get out very quickly, I sure want to be able to adapt to that, and I want to get oxygen delivered to my muscles and my arms and legs so I can get out of there and do that. Okay? Well, two other things that I'm going to talk about with respect to hemoglobin actually allow that to happen. Okay? The first is something called the Bohr effect. Okay? The Bohr effect. Now, this equation doesn't really tell you anything. Okay? It tells you that hemoglobin can hold on to oxygen. It tells you it can grab protons, and it can grab carbon dioxide, and it gives up oxygen. We just sort of knew that it gave up oxygen. This doesn't tell us anything that you wouldn't already otherwise know. Okay? This tells us a lot more. I'm going to explain it to you. For the moment, we're going to have myoglobin on there as a reference. Okay? What's the Bohr effect? Well, I'm going to tell you what it is, and then I'm going to explain this graph. The Bohr effect was observed by a scientist named Bohr about 100 years ago. This has been known for a long time. And what Bohr discovered was that if he changed the pH of a solution very slightly, that is, reducing it, putting just a little bit of acid into that solution, what he discovered was oxygen gave up more oxygen. Hemoglobin gave up more oxygen. Okay? Now that's what this graph is trying to show you. Here's physiological pH. pH let's say 7.4 is physiological. Right there, this blue line right here. Okay? That's that S-shaped curve that you saw before. Remember I said they would all converge up as we got up here? There they are converging. Okay? They're all converging up here. But look at the difference in these curves in this region right here, which is about, in this range in here, physiological oxygen conditions. Okay? Look at the difference. Lower means it's lower percentage saturation. Okay? So here's physiological. If we lower the pH from physiological down to here, we see that the very same hemoglobin will give up oxygen. If we lower it further, it gives up more oxygen. If we lower it further, it gives up even more oxygen. We see the pH going down from 7.4 to 7.2 to 6.8. If we go up, it holds on to it tighter. Yes?
Does that mean when we change the pH, the abilities of the hemoglobin to grab and catch the oxygen is reduced?
So when we change the pH, her question was, if the pH changes of hemoglobin, does that change its ability to grab and hold on to oxygen? Okay. Well, this isn't happening in the lungs, fortunately. Because if it were happening in the lungs, it would reduce the ability to grab a hold of that. This is happening out in the tissues that are exercising. Okay? Now, so you're actually getting just a little bit ahead of me. So let's talk about those tissues exercising. Okay? I have muscle cells that are exercising. I'm lifting weights, I'm jogging, which I like to do, or whatever that happens to be using muscles, I'm exercising. One of the byproducts of exercise is acid, making protons. Now, protons, therefore, are serving as a signal that, hey, I am going to need some oxygen here. That's what the muscles are flagging and saying. Okay? What's another byproduct of respiration, of actively respiring tissue? Well, carbon dioxide. Protons and carbon dioxide get released. That last equation that showed those was trying to tell you that, but this graph tells you better. Carbon dioxide will actually cause the hemoglobin to let go of oxygen as well. Okay? So, there's two signals that say release oxygen here. It's really favoring the release of oxygen for tissues that are going to need it, and that's not going to happen in your lungs because your lungs are not actively respiring like your muscles are. Does that make sense? Okay. This equation does show you one thing I haven't told you, though. Let's go back to the equation. The equation tells you that hemoglobin can bind to oxygen, and it tells you that hemoglobin can bind to these two things. Hemoglobin actually helps to carry carbon dioxide back to the lungs. Carbon dioxide is a poison, just like carbon monoxide is a poison. It's actually a very unpleasant poison. Carbon monoxide, if you have carbon monoxide poisoning, you fall asleep and presumably never know about it. If you have carbon dioxide poisoning, you get very toxic, icky effects. I haven't experienced either one of these, mind you, but of course, that's the case. All right. So, in the actively respiring tissue, we're making this. When we get back to the lungs, we go back and we do this. So the Bohr effect is explaining another important property about hemoglobin, how it delivers oxygen, and why it's working the way that it works. Questions about that? Yes? Yeah. So questions about sickle cell anemia. I'm going to talk about that in just a bit. So sickle cell anemia is actually a disease of hemoglobin, and there are some very interesting considerations for that, and I'm going to say something about that as well. Okay? Yeah, question?
Does the bending effect happen when carbon dioxide attaches to hemoglobin or just monoxide?
Good question. Does the bending effect happen when carbon dioxide binds to hemoglobin or just oxygen? It turns out that carbon dioxide binds to hemoglobin in a different place. It does not bind at the heme. And so this phenomenon of cooperativity that I've described to you does not occur with carbon dioxide, no. Okay? It could occur with carbon monoxide, okay? But not carbon dioxide. So carbon monoxide, remember, binds to the heme. Carbon dioxide is binding at a different place. Okay. These protons also help to change the structure of hemoglobin. If I start putting plus charges onto hemoglobin, I'm changing the distribution of charges that are on that molecule, right? And if we had a plus attracted to a minus, and I now put a plus on top of the minus, I've changed the nature of their interaction, right? Instead of being attracted to each other, they're not going to be attracted now. So we can imagine that adding protons to a protein will change its shape, and in fact, it does. Okay? I mentioned yesterday that we heat our food to cook it so we can kill the stuff that's in it because we're denaturing those proteins. Another way to denature proteins is add a lot of acid. Don't wash your hands with a lot of acid, okay? You're going to do some damage, okay? Yet another way we can denature proteins is by using soap. Detergents will also cause proteins to unfold and disrupt their function. We wash our hands because our hands are largely dead skin, right? So we don't care about the dead tissues that are on the surface, but we're killing the bacteria that are on the surface of our hands because that detergent is denaturing their proteins. Okay. Well, in this case, the denaturation is very tiny. It's not killing hemoglobin. It's only a very slight shape change, fortunately. Okay? Very slight shape change. Okay. Well, there's even more to hemoglobin's story. And the hemoglobin story gets even more interesting when we consider another molecule called 2,3-BPG. Yep, got the wrong thing there. Actually, no, I don't. This is right. I want to do this. Okay. This once again shows the structure of hemoglobin. Okay? Hemoglobin has 4 subunits, and maybe when I showed you this earlier, you might have said, hey, it looks kind of like a donut. Especially if it was this were morning and you were hungry, that might look like something. You might be thinking that. That's what I would be thinking anyway. Okay? That looks like a donut. Well, it does look like a donut. And it turns out the whole of this guy is really, really important. Okay? The whole of this donut is a perfect binding site for yet another molecule. Another molecule can bind right in the whole of the donut. The molecule that can bind there is called 2,3-BPG. How many of you have heard of 2,3-BPG before? Oop. Don't want to lose that? Oh, yours, okay. Somebody didn't want to lose it. I gave it away. Okay, nobody's heard of it. This is one of the reasons you take biochemistry. This is one of the most important molecules in your body, 2,3-BPG. We'll talk about it later in the term. But for right now, all you need to know about it is that just like protons and just like carbon dioxide, 2,3-BPG is a byproduct of rapid cellular metabolism. It's a third thing that's produced by actively respiring tissues. You don't need to know the structure of it. But the name, and it's a longer name, I won't tell you the longer name, but 2,3-BPG is a very important molecule. It binds in here. Well, what does it do? Okay. Well, let's imagine that I'm a hemoglobin, and I'm all loaded with oxygen. I've gone to the lungs, I've grabbed my oxygen, I'm leaving the lungs, and I'm going out to the tissues. Okay. Some of the hemoglobins, when they get to the tissue, say, oh, the concentration is lower, I'm going to let go of a little bit of oxygen. Right? If they all dumped oxygen at once, then we wouldn't get oxygen through the rest of our body. So this is a sort of a gradual process this is happening in. Okay? We get to actively respiring tissues, and the concentration of oxygen is lower. Hemoglobin drops some more oxygen, and some protons and carbon dioxide bind, and some more oxygen comes off. And this actively respiring tissue, in addition, is producing a molecule, or producing molecules of 2,3-BPG. 2,3-BPG binds into the whole of hemoglobin, and it causes hemoglobin to switch into what's called the T state. Hemoglobin actually exists in 2 different states. They're called T and R. T stands for tight. R stands for relaxed. Okay? Now, when 2,3-BPG binds, it converts hemoglobin into the T state. The tight state is a very low affinity for binding to oxygen state. When 2,3-BPG has bound to hemoglobin, that hemoglobin is pretty much going to dump its oxygen right there. It's not going to be anything partial about it. It's just going to dump its oxygen right there. Okay? So 2,3-BPG is helping to increase, again, even more oxygen being dumped where the tissues need it. Now, this hemoglobin now has passed through this tissue that needs the oxygen. It exits the tissue, and it's bound to 2,3-BPG. if 2,3-BPG is still going to be stuck to hemoglobin when the hemoglobin gets back to the lungs, you're going to be in trouble. Because it's going to be in the T state, and T state doesn't want to bind oxygen, and hemoglobin is going to get there and it's going to say, and it's just going to pass right on through. Well, fortunately, 2,3-BPG is not covalently bound, meaning it's a loose attachment. It comes on, it comes off, it comes on, it comes off. And some of the times, on the way back to the lungs, it comes off and a cell grabs the 2,3-BPG. Because 2,3-BPG has energy, and the cell can use that energy. Now that's really cool. Hemoglobin has dumped its oxygen. It's going to dump its 2,3-BPG. It's still in the T state. Remember I said when it got in the lungs it didn't want to bind oxygen? It took one to bind, then change that. When the hemoglobin gets back to the lungs, it's going to be in the T state, but that very first oxygen is going to bind, and we're going to see cooperativity, and it's going to change into the R state. It's going to grab all the others. The binding of the first oxygen causes hemoglobin to change from the T state into the R state. If there's 2,3-BPG stuck in there, it can't do that. Make sense? Now, the rather cool thing with this, not so cool if you're a smoker, is that smokers in their bloodstream have a lot more 2,3-BPG than non-smokers do. Oh yeah. So this means it's much more likely that hemoglobin of smokers is going to make it back to the lungs with 2,3-BPG in it. That means that smokers aren't going to be able to carry as much oxygen as non-smokers, and this is the reason why smokers get out of breath much more easily than non-smokers do. Their oxygen carrying capacity is lower because they have a lot more 2,3-BPG in their bloodstream as a byproduct of smoking. We'll talk later about why that's the case, but it's a byproduct of smoking. Okay? Another thing that smoking does here that reduces your oxygen carrying capacity is there's something found in cigarette smoke that I've already talked about. What is it? Carbon monoxide. Smoking increases levels of carbon monoxide, and so again, you're reducing the levels of oxygen being able to be carried by the hemoglobin because carbon monoxide is going and binding to the hemoglobin. Those two things are two very good reasons why you should not smoke. And of course, cancer is another one. Let alone it smells awful. But I feel sorry for people who smoke. I do. Okay, that's actually what 2,3-BPG looks like. It's a nice, tiny little molecule. You don't need to know that structure. It stands for 2,3-bisphosphoglycerate is what it stands for. I'll talk about it later. And this now shows curves if you have BPG and no BPG. It's exaggerated a little bit, but nonetheless, hemoglobin in the presence of BPG dumps its oxygen because the curve is lower. In the presence, it holds on to it. Okay, that's kind of exaggerated, but that's the phenomenon. Okay, fetal hemoglobin. All right, you thought you knew everything there was about about hemoglobin, but we have to have other considerations. The fetus needs oxygen just like mom needs oxygen. Okay, the fetus has to get its oxygen from mom, but if the fetus has the same hemoglobin that mom does, it's probably going to lose the battle because mom is going to hang on to it for the most part. The fetus is going to have a very low supply of oxygen if it has the same hemoglobin as mom does. Well, it turns out that the fetus has a very slightly different form of hemoglobin. We call it fetal hemoglobin, and what happens in the fetus is the two beta subunits aren't being made. They don't start being made until after you're born. Instead, they're replaced by two gamma subunits that are similar to the beta but not identical. So the fetal hemoglobin has a structure of alpha 2, gamma 2. Okay? The adult is alpha 2, beta 2. The fetus is alpha 2, gamma 2. Well, it turns out that the fetus, the fetal hemoglobin, has a greater affinity. That means this curve is above mom's hemoglobin. It has a greater affinity for oxygen than mom's hemoglobin does. That's really important because the fetus has to take oxygen away from mom's hemoglobin. If it can't do that, the fetus is not going to have very much oxygen. If we think about R states and T states, what it tells us is that the fetal hemoglobin is in the R state much more so than mom's is. Much more so than mom's is. There's a very good reason why that's the case. It's not just the slight structural change of the gamma subunits. I guess it is indirectly, okay? The gamma subunits, when they pair up with the two alphas, they make a very, very slightly tinier hole to the donut. The donut hole isn't as big. And since the donut hole was what was binding the 2,3-BPG, you guessed it, the fetus cannot bind to 2,3-BPG. What was causing the hemoglobin to switch into the T state? It was the 2,3-BPG. So the fetal hemoglobin stays in the R state. The R state likes to bind onto oxygen. That's how the fetus is actually able to take oxygen away from mom. Okay? One of the questions I commonly get, or I've even been known to ask questions on an exam, is, well, what if mom smokes? What's the effect on the fetal hemoglobin? Okay? The answer is there's no effect whatsoever on the fetal hemoglobin because it can't bind to 2,3-BPG. Okay? Mom can smoke all she wants. The fetus is going to have effects, definitely going to have some effects, because mom's going to be able to deliver less oxygen. That's a much bigger effect. But the fetal hemoglobin will not be affected by the 2,3-BPG. Okay? Many other reasons mom shouldn't smoke when the fetus is there, but 2,3-BPG is not one of them. Okay. All right. Now, the last thing I want to say something about is sickle cell anemia, because you asked the question over here about that. Okay? Hemoglobin is involved in so many things. Okay? We see it in our oxygen supply. We see it relative to a fetus. And we also see it relative to a very devastating disease known as sickle cell anemia. How many people here know somebody who has sickle cell anemia? Okay, yeah. It's a very debilitating disease. It's not fatal unless you overexert yourself, in which case it can be. Usually what happens is people who have it find that they get very, very weak and or very, very sore muscles when they try to do heavy exertion. Okay? Sickle cell anemia arises as a result of a mutation in one of the subunits of the hemoglobin. There's different types of sickle cell anemia and different types of mutation that cause that. But the most common ones that cause sickle cell anemia... I'm sorry? So alpha sickle cell anemia refers to a mutation in the alpha subunits. Okay? So the most common forms of sickle cell anemia involve a single change of a single amino acid in hemoglobin. The change of that single amino acid in hemoglobin causes the hemoglobin to polymerize under low oxygen conditions. It forms polymers, and polymers are long chains. Okay? So instead of having just a 4-protein subunit, I've got a 4 and 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 a 4, and they just go on and on. Okay? This happens during low oxygen conditions. Low oxygen conditions will occur when you are exerting yourself heavily because the muscles are using oxygen faster than the blood supply can deliver it. Okay? The reason that this turns out to be so painful is the muscle cells are literally starving for oxygen, number one. And number two, the polymerization of the hemoglobin changes the shape of the blood cell. The blood cell is a pretty little blobby little thing. Okay? Let's see. I don't have it on here. Okay. It's a pretty blobby little thing. Okay? Nice and rounded edges and so forth. And that's so that it can pass through your capillaries readily. Blood cells are nice and smooth for that very purpose. So they can pass through your capillaries very readily. Okay? When the polymerization of hemoglobin occurs, okay, that polymer causes the nice, smooth red blood cell to assume the shape of a sickle. It's very sharp-edged. That's where the disease gets its name from. And the consequence of that is that the red blood cell gets stuck in the capillaries. That stops the blood flow through the capillaries. And this is why the muscle cells starve for oxygen. They're getting nothing because they've stopped the blood flow through the capillary. The capillaries are where the most of the oxygen is exchanged. Okay? It's called anemia because the sickling of the blood changes the shape of the blood cell. And when blood cells get old, they get destroyed by the body when they're misshapen. That's what happens. When they get old, they lose their shape. So the body sees these sickle cells in the bloodstream, and it takes them out of action. So people who have sickle cell anemia have anemia. Anemia means low concentration of blood. And that's what's happening with sickle cell anemia. Now, there's a very interesting thing that's been discovered with sickle cell anemia in the past, probably 20 years. And that is that sickle cell anemia, it's been known, for example, that there are certain ethnic groups, racial groups that have a higher incidence of sickle cell anemia than others. And people have wondered, is it possible that there's an advantage to having sickle cell anemia? There's obviously some very important disadvantages to it. And what they discovered was there is a slight advantage to having sickle cell anemia. Sickle cell anemia, people who are heterozygous for sickle cell anemia have an increased survival for malaria, particularly when they're young. Heterozygous, meaning one mutant, one normal. Okay? If you have both, both mutations, you're in pretty bad shape. It's not going to give you any protection. It's going to hurt you. If you have heterozygous, you can live a fairly normal life, and you're actually protected against malaria. So it's thought that this is how and why sickle cell anemia has persisted in the human population. Okay? It has some obvious disadvantages, but things that persist for a long time probably have advantages as well, and this is one of them. Okay, I've given you guys an awful lot of stuff today. I actually do have a song that goes with this, so if you'd like to join me, please sing loud. Okay, I'd like to hear you sing loud today. It's a song that covers everything I've talked about today. It's called Hemoglobin's Moving Around. Oh, isn't it great what proteins can do, especially ones that bind to O2. Hemoglobin's moving around. Inside of the lungs, it picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively, thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration is high and has a role. Between the alpha betas, it finds a midazole. To empty their loads, the globins decrease. We need to bind 2,3-BPG. Hemoglobin's moving around. Get it down here? Here we go. The stages are set for binding a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and dumps the CO2 off to start all over again. So see how this works? You better expect to have to describe the Bohr effect. Hemoglobin's moving around. All right. See you guys tomorrow.